Puget Sound had the least healthy beach because we thought it would have lower oxygen levels and therefore lower biodiversity.
Well, we went 3 different beaches, Home, Whitney, and Big Beef Creek, which is Northwood Canal. We also went Shelton, which is South Puget Sound. Before we began our survey, we did a little research about each location. And we found that the Northwood Canal in general has lower oxygen level, the water quality is not good, lower pH, and it has lower dissolved oxygen. The water in the canal is cold and lacks nutrients. Also, for both South Puget Sound and Northwood Canal, there's aquatic. For both Puget Sound and Northwood Canal, there is a lot of ongoing logging system. Also, land development practices. Also, there's contamination streams. It is affecting the environmental condition. Also, the landscape is changing from native cover to urban due to people moving and settling down the coastal areas. That's what we found out. Also, we visited each places and we basically did the beach survey. We basically set up a transact line and we identified the different species found on the beach. Also, we counted and basically recorded.
On each beach, we collected 20 oysters that we took back. We sampled their gills and took back to the lab to do gene expression tests on them.
So why oyster gills? Oysters are good filter feeders and they have good bioindicators because they simply sit in one spot. They simply sit in one spot and take on whatever happens to be in the water. For example, if there is a toxin in the water, the oyster can filter the water through their feathered gills and removing the pollution and the toxin. So by examining their gill, you can understand a lot about the water, also the habitat they're living.
So we looked at gene expression because it changes based on the environment. So when a change happens in the environment, it cues a change in the oyster itself. Gene expression tells the cells to produce more or less of a certain protein, and those proteins translate to a change in the organism's body.
Basically, gene expression refers to the process of DNA being transcribed into RNA, and then the RNA is converted to make a specific protein. So these expression of proteins change to deal with different things. For example, if you get a cut, your body produces more antibodies to heal the cut.
So these are our methods. We looked at environmental data that was available online. We laid out a beach transact line and did the survey, which would show us biodiversity. And we used gene expression from the 20 oysters we collected from each beach.
This is the environmental website we use. It's called NANUS. They provide salinity, temperature, sea level, oxygen level. Basically, they monitor ocean and basically northwestern region. This is where we got our data information.
At each beach, we laid out transact lines. We found a reference point on each beach, and with this, we laid out a line from that reference point to the end of the beach. On the line, we found plots, and within the plots, we found random points to look at. We used the Shannon Diversity Index with the data we found in each random quadrat or area in the plot to measure the richness and evenness of the species on the beach. From each beach, we collected 20 oysters, and we took them back to the lab to do tests on them.
Our steps were, first we isolated the RNA.
This was extracting RNA from the oyster sample of the gill.
Then we DNA'd the RNA.
This was making sure we only had RNA in our sample and no DNA.
Then we reverse transcribed the DNA
turning it into cDNA and creating more of a strong copy of it so we could do our final step.
Then we did qPCR.
This step was putting it in a machine and analyzing what was in there so we could look at how they differ. The two gene expressions we looked at were HSP90 and CYP1A1. HSP90 stands for heat shock protein, and CYP1A1 stands for cytochrome P450. HSP90 deals with certain stresses on the cells, and CYP1A1 deals with detoxification and pollutants in the cells. These are our results.
As you can see, Northwood Canal has lower oxygen level, but both South Puget Sound and Northwood Canal have a healthy oxygen level. Also for chlorophyll, as you can see, Northwood Canal lacks chlorophyll, and South Puget Sound has a fair amount of chlorophyll.
Salinity is the amount of salt that is in the water, and South Puget Sound has higher levels of salinity, while Northwood Canal has more varying and lower levels of salinity. Temperature-wise, South Puget Sound has lower but more varying levels, and Northwood Canal has higher levels of temperature. We decided that South Puget Sound had better quality, it had higher chance of things living there because of its better quality in the water. This was our biodiversity results that we found using the Shannon Diversity Index. As you can see on Point Whitney, it had its lowest levels of biodiversity at the beginning of the beach, and towards the end of the beach, it had its spikes in biodiversity. And you can see how that shows the better beaches. We went to Shelton and Big Beach. Shelton had more oysters, which we found correlated with higher levels of biodiversity. And we think this is because the oysters provided habitat for other animals to live in.
One of the genes we did was HSP90. As you can see, HSP90 is expressed more at Point Windy and least at Big Beef Creek. Our result shows that something in the water at Point Windy was causing the oyster cell to stress. Because of these, the oysters were making more HSP90 as a stress response. HSP90 is the heat shock response. Basically, it is caused by a lot of things like environmental conditions, exposure to toxin or hypoxia. As you can see, Shelton, the Point Wendy didn't have like a lot of salinity level was low, the oxygen level was low. Maybe that's one of the factors that contributed to this.
These are the results for the CYP1A1 gene expression. Big Beef Creek had really high stress levels on that. As you can see, Big Beef Creek and Point Whitney had really high stress levels, but they were caused by different things.
So overall, the Shelton had the bigger oyster than all the other big places.
Some of the possible reasons that the oysters were bigger at Shelton were because it was an oyster farm for the first thing, and Big Beef Creek and Northwood Canal, the beaches were both public beaches that people had previously harvested oysters from.
And not a lot of nutrients in the water. Maybe that would be one of the things.
So this is our beach report card that you can see we rated each of the beaches based on biodiversity, the stress levels of HSP90, and and the environmental data.
So from looking at the report card, Shelton had better beach health than for Wendy and
In conclusion, I learned that high biodiversity doesn't necessarily mean a healthier beach. It's important to look at a lot of variables when you're measuring something as big as beach health.
Also, gene expression, stress is related to related gene corresponds to the environmental data. Like global warming, you basically know it is affecting the species who live in the water. Is it like good? Most of the species don't have like enough oxygen. Like the salinity is going down, which is affecting them. So that would be one of the better. So from like looking at the environmental data, you could assume that the animals are being affected by the environmental change.
We want to thank Emma,